Good evening to one and all. My name is Madhukar. I am studying BSc BZC first year TTWRDC mains both at ADILABAD. Today my topic is structure of bacterial cell. This is the structure of the bacterial cell. Next one, context, introduction, cell morphology, structure of a bacterial cell, cell wall, plasma membrane, extracellular, external structure, intracellular, internal structure. First of all, we are telling about this. Introduction. All bacteria are unicellular organisms that are responsible by binary fusion. Most of bacteria are capable of independent metabolic existence and growth, but species of Chlamydia and Rickettsia are obligatory intracellular organisms. The bacteria cells are extremely small, are most conveniently measured in microns, 10.6 meters. The bacteria cells are usually between 0.4 and 1.5 microns meter in short diameter. Next one, cell morphology. Bacteria have a characteristic like shape. The common microscopic morphologies are spherical or ovoid, is corky. Rod shape is bacilli. Comma shape is vibrio, and spiral shape spirillum and spirocheta. Some corky characteristically grouped in pairs or chains. Some form grape-like clusters of special cells. Some round corky from cubic factor. Bacteria or other specially grow separately. The microscopic reference is therefore valuable in classification and diagnosis. Next one is the structures of a coccus, round shape, bacillus, rod shape, streptococcus is chain shape, diplococcus is pear shape, spirocheta is spiral shape. And next one is structure of a bacterial cell. The protoplast, the whole body of living material. Protoplasm is bounded peripherally by a very thin elasticity and semi-permeable cytoplasm membrane. Outside and closely covering this lie the rigid supporting cell and relative permeability. Next one is the cell wall. The cell wall encases the protoplast and lie immediately external to the cytoplasmic membrane. It is 10.25 nanometer thick, strong, and elasticity rigid through with some elasticity and openly porous, smaller than 10 kDa in mass and 1 nanometer in diameter. It shows granule structure and lacks microfibrils. Gram positive and gram negative bacteria have a different types of bacterial cells. And next one is this is the structure of a cell wall. Next one is the gram positive cell wall. The gram positive type wall is relatively thick, about 30 to 100 nanometer, and it generally has a simple uniform appearance under the electron microscope. Some 40 to 80% of a walls is made of complex polymer peptidoglycan. In this type of wall, succulents consist of a multilayered peptidoglycan which during growth developed by the inside to outside mechanisms. Next, covalently bond of peptidoglycan compounds such as teachers acid, typical substituted polymers, glycerol, phosphate, repeater phosphate. In some bacteria, Mycobacterium, the wall contains lipid, while in other strain of a Streptococcus, it contains carbohydrates. The position of a wall can vary with growth condition. For example, Bacilla, the availability of phosphate affect the amount of a cell wall teaches acid. Next one is the gram-positive cell wall. This is the diagram of a gram-positive cell wall. We know that. And next one is the gram-negative cell wall. The cell wall of a gram-negative bacteria are more chemical complex, thinner and less compact. Peptidoglycan makes upon only 5 to 20% of the wall and is not a outermost layer. The peptidoglycan of gram-negative bacteria is located between the plasma membrane and of our LPS membrane. This LPS membrane is similar to the plasma membrane but is permeable and is composed of liposaccharides, LPS. And next one is gram-negative cell. This is the structure of a gram-negative cell. Plasma membrane. Next one is a plasma membrane. Plasma membrane in bacteria are composed of a phospholipids containing a polar group attached to a 3 carbons glycerol backbone. There are also 2 fatty acid chains dangling from the other carbons of a glycerol. The phosphate in a molecule hydrophilic is attached to the water. The fatty acids are hydrophobic. Membrane also contains proteins. Typically 20 to 30% of a membrane, most of them are placed in the membrane. So the hydrophobic ammonia acid associated with the lipids in the membrane and the hydrophobic amino acid are outside membrane interacting with either the cytoplasm. Functions of the plasma membrane is selectively permeable in that help control what moves into the outside. Next one is the extracellular structure. Fimbriae or villi. Fimbriae or villi are elongated or hair-like proteinaceous structure which proteins from the cell surface. They are especially on this gram-negative cell. Fimbriae are typically 2.3 nanometer in diameter from 0.1 micrometer to several micrometer in length. They may occur over the cell or may be localized. And next one is the flagella. The flagella made up of a class of linear protein called flagellants. Flagella which arise at various points on cell surface. Flagellated bacteria are described as monotrichous, lopotrichous, or unpeptitrichous. It is depend on how the flagella are arranged. Capsule and slime layer. The capsule of most bacteria consists of a polysaccharides. The bacteria single species can be classified into different capsules. Several types based on the fine chemical structure. These polysaccharides. Intracellular structure. Plasmid. The term plasmid was first introduced by American molecular biologist John Lederberg in 1952. A plasmid is a short, usually circular and double-stranded segment of DNA that is in the cytoplasm, separate from the main bacteria chromosomes. Their size varies from 1 kilo base pair to over the 100 kilo base pairs. And next one is the plasmid are capable to autonomous replication. Plasmid can transfer gene from one cell to another cell. And next one is the ribosomes. The cytoplasm contains a large number of solute, low and high molecular weight substance RNA, approximately 2000 ribosomes. Bacteria have 70 ribosomes comprising 30s and 50s subunits. Functions of a ribosome. Ribosomes are the organelles for protein synthesis. And next one is the mesosomes. Mesosomes are convoluted or multinucleate membrane bodies visible in the electron's microscope. They develop by complex integration the cytosol membrane into cytoplasm. Functions of a mesosomes. Mesosomes functions of a mesosome. Function is in the compartment of a DNA at the division and sporulation, having a function elongation to the eukaryotic cell, providing a membranous support for respiration enzyme. This is nucleus. The nucleus is a region of cytoplasm where the chromosomal DNA located. It is not a membrane bonded molecules and simple an area of a cytoplasm where the standard of a DNA are found. Most of bacteria have a single circular standard chromosomes that is responsible for replication. Although a few species do or have a two or more smaller circular auxiliary DNA strands called plasmid are also found in cytoplasm. So thank you watching this video.